If you dive deep enough into your own bloodstream, you enter a completely alien environment. Down here, it's an active, invisible war between the cells keeping you alive and the invaders trying to tear them apart. For most of human history, fighting those viral invaders has been a game of chance. We either wait for a natural immune response to kick in, or we flood the body with blunt force chemicals and hope the treatment kills the disease before it hurts the patient. But imagine removing chance from the equation entirely. Imagine deploying a microscopic, programmable assassin that navigates directly to a threat and neutralizes it exactly where it hides. Shifting from a broad defensive biological reaction to a targeted, programmable offense changes the math of how we survive a disease. Scientists are already building these programmable microscopic robots, but they aren't using silicon, gears, or metal. They are constructing them entirely out of DNA. In early-stage experiments, researchers are programming these DNA machines to hunt down specific organic targets, like cancerous cells or the SARS-CoV-2 virus, right at the atomic level. Repurposing the building blocks of life into programmable machinery bridges the gap between organic biology and mechanical engineering. You can't just take a traditional robot, shrink it down, and expect it to work. At the macro scale, physics is predictable, but at the nanoscale, the fluid dynamics are completely chaotic. The biggest obstacle is a phenomenon called Brownian motion. At this size, water feels as thick as molasses, and surrounding molecules constantly vibrate and crash violently into everything around them. If you built a tiny motor with traditional rigid metal joints, the extreme friction and relentless physical battering from that Brownian motion would tear the machine apart almost instantly. To survive this environment, scientists use folding techniques inspired by origami. They weave flexible strands of DNA over and under each other to create rigid structural joints that can absorb and withstand those chaotic vibrations. At this scale, engineering success is defined by how well a structure can endure these constant, high-energy molecular collisions. Since mechanical motors fail here, researchers have to hack the chemistry. This animated diagram shows a biochemical process called DNA strand displacement. Notice how these two complementary vector strands of DNA are exchanging geometric segments. One segment acts as the physical structure, and the other acts as the fuel. As that specific fuel sequence attaches, the biochemical reaction forces the folded origami structure to change its shape. This is how scientists program the robot to take a step or extend an arm with sub-nanometer accuracy. Chemistry handles the motor, but we still need a steering wheel. Because the molecular environment is always shifting, researchers apply external electric fields to push and guide these robots from the outside. They also use localized flashes of light and magnetic waves. By manipulating these physical signals, scientists can direct exactly where the DNA machines move, adding a highly accurate layer of secondary navigation. Once the robot is steered to its destination, it deploys its payload. The folded structure identifies the unique chemical markers of a disease cell and physically locks onto it, snapping its microscopic claw shut around the target. By combining internal chemical fuel with external physical fields, researchers have created a fully steerable molecular computing platform. Right now, these machines are in the proof of concept phase. They are simple designs operating in isolated laboratory environments, rather than complex fleets navigating the bloodstream of a living patient. Moving from the lab to the real world is difficult because of major knowledge gaps. We don't yet have comprehensive databases that map out the exact mechanical properties of folded DNA structures. To solve this, scientists are combining artificial intelligence simulation tools with standardized DNA parts libraries. AI helps predict how specific DNA shapes will behave, accelerating the design and biomanufacturing process. Reaching real-world healthcare applications requires treating biology like a standardized manufacturing supply chain, and that manufacturing potential goes far beyond medicine. These robots can act as programmable templates for advanced construction at the sub-nanometer scale. Theoretically, a fleet of DNA robots could position individual nanoparticles one by one. This precision would allow us to build highly efficient optical devices and next-generation data storage systems. For our entire history, we've built technology by extracting and molding static materials like iron, plastic, and silicon. Now, we are learning how to grow intelligent machinery biologically. Applying the logic of programming to DNA transforms biological building blocks into a functional material for the nanoscale. This provides a new path for building at the scale of a single molecule.